My name is Kika Sucharov, and I am one of the co-chairs of the Young Investigator Award competition. And I'm here with Dr. Castro, who is one of the finalists and is going to tell us about her work.
Hi, I'm really happy to be here and to be able to discuss of my work, and really glad to have the chance to present my work tomorrow.
So tell us about your science and how you came to your question.
Sure. So my work is about atrial fibrillation, and we start this work in my lab with a GWAT that has been done in collaboration with the Dr. Elinor Labs, where we found a new association of activin receptor type 2A with atrial fibrillation. And in atrial fibrillation, I think there is a lot of research that try to understand better the mechanism, but there is still a lot of gaps in the pathophysiology and some limitation in development of new drugs. So with this new association, we start this project trying to understand what is the specific role of this receptor in AF, with hope maybe that can lead to new therapeutic option.
And where are you now with the project?
My work is mainly basic science. I did an in vivo model and in vitro model. So we wanted first to validate that these actors can lead to atrial fibrillation. So we test that in different animal models, and we validate that just the activation of this pathway can lead to atrial fibrillation. And then we wanted to modulate this pathway. And one of the things is there is two activin receptor type 2, a B and an A, and so we wanted to see the specificity of this receptor, because during a long time people were thinking that these two receptors play the same role, and so it's nice when you have something really specific that it's like give you hope that it will work for maybe therapeutics. And so then we play on these two receptors to really see the specificity, and doing knockout, also using some inhibitors. Especially now there is a lot of development of ligand trap for the receptor 2A or 2B. And so with this use of drugs and knockout, we were able to see a specificity of this receptor in the induction of atrial fibrillation. And so that was just validation of this receptor. And then I did more in vitro experiment in cardiomyocyte, where I was trying to understand the pathway leading to this atrial fibrillation, and we discovered that this receptor can lead to an increase of ubiquitin ligase, Smurf1, and that Smurf1 can decrease the expression of Connexin43 that is well known to participate to atrial fibrillation pathophysiology. And in vivo model, we're able that we can shortcut the pathway of activin receptor 2A by maintaining a level of Connexin43, and so we'll reduce AF inducibility.
So do you think that in terms of a next step of pharmacological inhibitors, would you see targeting Smurf1 or the receptor?
The idea now is probably to try the receptor with all the ongoing clinical trials of this ligand trap, specifically for 2A and 2B. Last year, one activin receptor 2A ligand trap was received FDA approval. Was not in atrial fibrillation, but he was on pulmonary arterial hypertension. So I think it gave us hope that this can be a transition to atrial fibrillation. But I think all the pathway could be interesting, but the one that seems close to maybe be possible to transition to clinic is maybe activin receptor 2A target.
Okay. Very interesting. I wish you
you.
good luck.
Thank you so
That is an
much.
important problem.
Yeah, thank you.
Thank you very much for your time, and I look forward to your presentation tomorrow.
Thank you.